Okay, so last chapter is chapter 16, Studying and Manipulating Genomes. This talks about the methods in which the genomes and DNA are manipulated and altered. Okay. Cloning DNA. So what is cloning DNA? Cloning DNA is taking one small fragment of DNA, a gene, a part of a gene, several genes, depends on what you're doing. There'd be no genes at all, just some DNA. And you make an exact copy of that piece of DNA. Now, cloning, you might have, think of the famous cloning where, you're making a perfect duplicate. That is not the case. You're just making a copy of a stretch of DNA. You're not copying an entire organism. This is not Star Wars, an Attack of the Clones. Nothing like that. You're just taking a piece of DNA and making a copy of it. That's it. We use, in doing this sometimes, not always, restriction enzymes. So what are restriction enzymes? These are enzymes that will target a particular sequence of DNA, go in there and chop it up. That's what a restriction enzyme is. It's kind of like a bacterial immune system, in a sense. It will go in and find a particular DNA, in this case foreign DNA, and chop it up. Now, we found this to be very useful because it's site-specific. It will only make a cut at a particular sequence of DNA. When it cuts DNA, many times it will leave what we call a sticky end, where there's an overhang of bases on each end. Now, this is handy because we can actually use that to clone DNA. You also need to know what recombinant DNA is. Recombinant DNA is DNA from two or more organisms. Okay, for example, I might take bacterial DNA and add it to human DNA. Put that in a bacteria. That is recombinant DNA. Now, we use this to clone DNA. So what do? Let's just say we want to get a copy of one gene. So we'll use restriction enzymes, that's panel A, and we'll cut the DNA. And we're going to cut before the gene and after that gene. Then we'll take that enzyme and we'll cut into a plasmid, which is a circular piece of DNA. We then combine these fragments in panel C and D, and we make one circular piece of DNA that is recombinant DNA. That has the plasmid, which allows this to replicate in a bacterium, and it also has our DNA of interest, which is part C. These then combine to make a vector and an insert, and we can then add that to bacteria. Another strategy for cloning is cDNA, or complementary DNA. Now, we've talked about how RNA is processed, right? That DNA is made into RNA, and then RNA is cut up and spliced together. Well, what complementary DNA does, it allows us to clone that processed RNA. Okay, we might want to look at a gene product, a protein, and we want to see the final sequence of it in DNA. If we went to the gene, well, that won't work because it's got the introns and the exons, and it's kind of interspaced. So what we do is we collect the RNA. When we collect the RNA, what do? We then make a copy of that RNA as DNA called cDNA, and then we then clone that. Okay, so let me show you how that works. So we take RNA and we use an enzyme called reverse transcriptase. This is going to make DNA from an RNA template. That's the second part. We then get rid of that RNA. Okay, that's the third and fourth part, and replace that RNA with a complement of DNA. So you see what we did? We made double-stranded DNA from that RNA at the top. Okay, so the top is RNA and the bottom is double-stranded DNA that's made using RNA as a template. And this is very useful when you're looking for not the gene, but the processed form of the gene, because RNA undergoes transcription, it undergoes the processing and removing of introns. Okay. DNA libraries. What are DNA libraries? Well, what's a library? It's a place that has lots of books, right? Well, if we want to have all the genome of one organism, do we want to have that whole genome in one big book? No. No, it'd be a book that was like, 50 feet long. So what we do is we chop out different sections, and each section is like a volume of a book. And those volumes together represent the genome of an organism. So think of it like an encyclopedia. What's an encyclopedia? It's got several smaller books, all related to one field, like general knowledge. Well, a DNA library is kind of like that. Each book is a chunk of an organism's genome, and it's categorized, and it overlaps, and it allows us to keep the whole genome of an organism in separate, discrete parts. We're not going to worry about probes. Okay, PCR. You should know a lot about PCR. Very important technique. So what is PCR? Polymerase chain reaction. Simply put, we're going to use primers, and we're going to take one strand of DNA, or a few strands of DNA, and make a lot of DNA as a result. Okay. You need to know the different parts of PCR. Okay. What are the different steps? First, you separate the DNA, or denature the DNA. Second, you attach the primers. And then third, you extend. So let's just look at this diagram above here. Okay. So part A, we have our double-stranded DNA, and that's blue. Part B is we heat the DNA, and we break those hydrogen bonds. As we break those hydrogen bonds, it allows our RNA or our DNA primers to sit on the DNA, those red strands. And those are going to serve as the attachment point for polymerase. Part C is that polymerase will sit on the DNA at the primer and move away from the primer, move away from that red band. And as they move away, they're going to add nucleotides to make it double-stranded. So what do we have here? We have one strand of DNA. We separate that one strand, and we make two separate molecules of DNA, and then we use those as templates, and we make two double-stranded molecules of DNA. So we go from one DNA molecule to two DNA molecules. Now, in the process of PCR, this is an additive process, meaning that one will become two, two will become four, four becomes eight, eight becomes 16. It's an exponential cascade reaction. Now, when we do PCR in the lab, we'll do 30 cycles, 40 cycles, 50 cycles in some cases. And each cycle is doubling it. Now, initially, you hear the numbers like 2, 4, 8. It doesn't sound like a lot. But when you start doing that for 10 cycles, 20 cycles, 30 cycles, you're into trillions and quadrillions of different copies because it's this exponential growth. Because what do you need to know about PCR? Well, the steps of PCR we went over in class. And also, what does PCR do? It makes a lot of DNA from a few copies. Why is this useful? Well, crime scene DNA, for example. You can use PCR to amplify very small amounts of DNA from a crime scene. Many other uses we're not going to go over, but it's one of the primary techniques that we use in biology. This kind of goes over the steps, and you might want to know the steps. We have denaturation, where we heat it. We have primer annealing, where those primers sit on it, part B. And then C, we have extension. The polymerase extends outward. And that is all we're going to cover. There was some other stuff on genomics and DNA chips. We're not going to worry about that. We're just going to leave it at PCR. Okay, and I hope that helps, and good luck on your test.